I'm Anna Henry, and I did my thesis on the morphologic examination of isolated vascular smooth muscle cells cultured under shear stress using a novel bioreactor system. So what does that mean in words that everyone can understand? We looked at fluid flow in blood vessels, more specifically how it affected the smooth muscle cells. So my advisors were Dr. Wilkins from the chemical engineering department and Dr. Crane from the biology department, and it was a wonderful experience to be able to work with both departments and have a joint project. So how does this actually matter? We're looking at coronary artery bypass grafts. So that's when we take a vein or an artery from a different part of the body and use it to bypass a blockage in the heart and reestablish blood flow and therefore oxygen flow to ischemic parts of the heart. So they've noticed that when they're using these grafts, then they have both venous and arterial grafts, that the venous grafts have a lower patency, and the question is why? So venous graft failure happens through a mechanism called intimal hyperplasia, which is where you get a thickening of the vessel wall that results in occlusion of the blood vessel. So it gets so thick, and as you can see with the vein here, that the wall is much thinner than the arterial wall. So that venous wall thickens and thickens and thickens until you actually completely block the blood vessel. So there's a couple of differences between conditions, arterial conditions and venous conditions that may affect this, and they are compositional related through the partial pressure of oxygen, because you have oxygenated blood in the arteries and non-oxygenated blood in the veins, and flow related conditions including normal stress, which is where you have the fluid pushing on the walls because of pressure, and that expands the arteries this way, and you also have a shear stress. So shear stress is what we're interested in. And you may ask, what is shear stress? So shear stress is a parallel force along the wall that's caused by the flowing fluid. So think about if you have water flowing over your hand. You can feel that water pulling on your hand and dragging on your hand, and you know that that's the force that's applied. So shear stress is a function of several variables, including the vessel radius. So how large the vessel is impacts the velocity of the fluid that's flowing through it, and therefore the shear stress. The viscosity, think if you have gel flowing over your hand, it's going to have more drag or more pull, whereas if you had water, it would have less. So increased viscosity would increase shear stress. And then your flow patterns also affect the shear stress. So an increase in velocity, if you have fast flowing water, it's going to pull harder, and if you have the slow flowing water, and the way that fluid interacts with the wall also impacts the stress. So we're looking at smooth muscle cells, and you may ask, why are we looking at smooth muscle cells? So smooth muscle cells, as you can see, there's a cross-section of an artery, and you can look at, there's the endothelial layer, the smooth muscle layer, and the fibroblasts. So the smooth muscle layer is involved in intimal hyperplasia and is that layer that gets thicker in graft failure. And it is separated from the flowing fluid generally by the endothelial cells. So that is also impacted by the shear stress. And Ellen, who's working in our lab, was looking at endothelial cells. So we're looking at if we excise those smooth muscle cells from the vessels, will they respond similarly when they're removed from the other tissues, and also when they're removed from their three-dimensional structure. So if they're just laid flat, will they also respond to shear stress? So what are our expectations? Smooth muscle cells are adherent cells, which means that they hold on to whatever you put them on, assuming they're able to. And they do change under flow. The literature has shown that when you expose smooth muscle cells to flow, that they line up with the flow, and they elongate so that they form more of a tissue layer. So we're also expecting that when we see those, the cellular elongation and alignment, that we're also going to see subcellular structure remodeling of the F-actin. So F-actin is basically like the cellular skeleton. You can see it here, it's all these little dots on the cell, and it enables the cell to basically change structure. So how are we going to look at this? Well, we are examining visually, using microscopy, how the cells are changing based on the shear stress. And we're looking at morphologic changes, so general shape changes of the cell. Are they getting longer? Are they lining up with what's around them? And then subcellularly, we're looking at how the F-actin filaments are aligning based on using a phalloidin stain that is the red you can see here, that attaches to the F-actin and allows them to glow a red color. And then we're addressing how the nuclei, we're looking at the nuclei using a DAPI stain that enables us, which is the blue you can see here, that enables us to see how the actin is changing around the nuclei and whether we have the cells that we think we have later on. So our hypothesis and objectives, we think that the application of arterial shear stress should result in alignment of our smooth muscle cells and remodeling in the direction of the flow, both on the cellular and the subcellular level. And then we are looking to evaluate the effects of shear stress on the vascular smooth muscle morphology, and we're also looking at whether there's a difference between venous smooth muscle cells and arterial smooth muscle cells of the iliac artery and the iliac vein, respectively. So before we can look at how the shear stress affects it, we need a way to apply the shear stress. So we developed a bioreactor, and these are some of the requirements that we were thinking about when we were developing our bioreactor. We need to be able to apply a controlled shear, which means a controlled velocity and a controlled radius. We need to be able to fully develop the flow so that we can characterize the shear stress that we're applying. We need to have an adequate growth area for our cells so that they can sit down and they can grow and hopefully be happy in the environment that they are and be able to express the way they would normally. We need to allow for cell attachment, and then again, making sure that we have those optimal, or trying to make sure that we have those optimal growth conditions to make them as happy as possible during the experiment. So this is our reactor flow system. What we came up with, as you can see, it's a closed loop. So we have a reservoir, a slide for applying shear stress, and then the pump that provides the fluid motion. So our pump, it provides a range of fluid flow rates and is a pulsatile pump, so it more closely mimics the physiological conditions. Our tubing is autoclavable, so we can provide a sterile system for our cells. Our reservoir is the right size, and it minimizes the waste and allows us for an extended culture. And our slide provides our growth surface, enables us to define the shear stress that we're applying, and is clear, so it allows for microscopy. So shear stress application, how are we actually going to go about doing this? As we said before, our shear stress is a function of the flow rate and the viscosity of the media. And a previous thesis student looked at the viscosity of the media and was able to define that for us. So our system, based on the equation down here that was provided by the company, allows us to see that we can apply shear stress between 18 and 64 dienes per centimeter squared. So arterial shear is right around 16 dienes per centimeter squared. So as you can see, we have quite a bit of flexibility and are able to apply that level of stress. To give you a better idea of exactly what we're doing, we're applying porcine gelatin to the base of the slide to allow our cells to have something to attach to. We're applying our cells in a confluent layer so that they are as close to the tissue setup that they have in the body. And then we're allowing our cells to attach for several hours so they can sit down and resist that shear stress. We're applying the shear stress for a 28-hour period at 16 dienes per centimeter squared, which is the arterial shear stress. And then we're looking, using our fluorescent staining and microscopy that we talked about earlier, to see how those changes actually occur. So these are the results that we saw for our static arterial and venous smooth muscle cells. And you can see that there isn't really any alignment between the cells, which is what we expect. You can see that the actin is going basically every which way, and that the cells do appear to be in a confluent layer. So when we applied the flow, you can see there are definitely some changes. So you can see the beginnings of alignment as things are starting to sort of move in the direction of realign, in the direction of the flow. As you can see, the flow is going from left to right. You can also see that as they're beginning to remodel, you're getting a change in the cell shape. So when the artery, especially in the arterial cells, you can see that the F-actin is kind of collapsed, and we would expect over a longer duration that it would sit down and have a more natural shape to it. As you can see in the venous cells, they are still attached, or maybe they have reattached. Something that we're looking into. You can also see that there's a decrease in the cell density. So when that shear stress is applied, we are having fewer cells that are able to survive that and retain their attachment. So as we saw, there were definitely some cellular changes as a result of shear stress application. There's a decrease in cell density. We have fewer cells on the plate, and we're hypothesizing that the shear stress is causing those cells to lift, and that's why we're getting that decrease in density. It seems like the venous cells, as you can see, are possibly more resilient to shear stress. There are many more of them still present, and they do appear to still have that physiologically relevant conformation. So it's possible that they may be able to tolerate higher levels or higher changes in shear stress, greater changes. And you can see that we're beginning to see the alignment of the cells and their subcellular structure under flow. Looking forward and future work-wise, we're hoping to optimize the growth conditions further. We maintained our system at 37 degrees C, but we were not able to apply 5% CO2 that's optimal for cell growth because we were outside the incubator. We'd like to further examine the flow losses, or the cell density losses as a result of flow application, and have further comparisons between venous and arterial cells. We'd like to look at different levels of shear stress in venous conditions. So if you put in venous cells under venous conditions, do they behave differently than arterial cells under arterial conditions or arterial cells under venous conditions? And also to extend that duration. So we looked at 28 hours. As you can see, the cells had clumped up, but they hadn't quite extended yet. So hopefully with a longer culture time, you would see those cells re-extend, reattach, and actually see that directional alignment. I'd like to thank my advisors, Dr. Wilkins and Dr. Crane. I couldn't have done it without them. It's been quite an experience being able to work with both the engineering department and the biology department. My colleagues and partners in crime, Venki and Ellen, we worked through lots of struggles and also celebrated lots of triumphs. So it's been quite a year in the lab. And my family and friends for supporting me through this process and for listening to all of my talk of cells and research. So thank you to the University Honors Program for providing the funding for this project, and thank you to all of you for coming. So are there any questions? Oh, these are my sources. And are there any questions?
So I noticed you used a porcine gelatin, and I believe the cells you said were human cells.
Correct.
Right? Was there any issue with compatibility that you noticed?
No. The gelatin, basically, think of it like a jello layer sort of thing. So it's a micro layer, and it basically just provides them a soft surface to sink their little feelers into so that they're able to really hold down.
How different is the venous flow to the arterial flow? You said you
So
want to compare the different
venous flow
types.
physiologically?
Yeah.
So physiologically, when you think about an artery, you have that high pressure
Right.
versus venous where you have low pressure. You also have constant flow in an artery in that pulsatile sense, where in the venous flow, you're not necessarily seeing that same pulsatile flow when you're using a different pump because of the skeletal muscles relate into that. So you expect a much, much lower shear stress because the blood is not flowing as fast, and because it's moving uphill instead of downhill for the most part. So you would see a lower shear stress, and as you can see in the structures that they don't need to withstand that same pressure, that same fluid interaction. Yes.
You had said that the cells were affected by both shear and normal stresses,
Right.
and you only focused on shear stresses.
Exactly.
Do the normal stresses not play as big a role, or?
Well, so we're looking at one. We were trying to isolate that variable in order to determine if that's a possibility. So obviously, if we were able to continue and look at multiple variables, we could examine that partial pressure of oxygen, examine that normal stress, and see, okay, is that one of the contributors? But in order to isolate the shear stress and say, okay, can we rule out shear stress as part of as a situation we focus on shear stress alone? So it's very possible that normal stress is part of it, and there are other labs at other universities that are looking at how normal stress affects cell growth.
There's no further questions. Thank you all for attending.
Thank you.